Hi guys, Miss Francis here to talk to you today about DNA, the molecule of life. We're going to get into the details of DNA. This molecule was introduced during biochem, so you should have a nice foundation upon which we can build this information. So Mendel and Morgan, they didn't know about DNA, they didn't know about genes, they didn't know about chromosomes. They called these things factors. They didn't know the what that was controlling these different patterns of inheritance. So the next group of scientists really sought out to figure out the what. What is it that's causing these different patterns of inheritance? And there are some key scientists. They include Griffith, Avery, Hersey and Chase, Chargaff, Watson and Crick, Wilkins and Franklin, as well as Linus Pauling. So at first, scientists actually believed that proteins were the genetic material, and these scientists sought to figure out, is protein in fact the genetic material? Is that in fact the factor that Mendel was discussing? Well, research began with Frederick Griffith. And what he did was an experiment using a strain of bacteria. And he took two different strains, the R strain, which is sometimes called R cells, and the S strain, which is sometimes called S cells. And this is what he did. The S cells, or the S strain, have a capsule. So here is my S strain. They have a capsule. That capsule protects them from the mouse's defense system, and as a result, the mouse dies. The F strain, or the F cell, the R strain, or the R cells, do not have a capsule. So that leaves them vulnerable to the mouse's defense system, and the mouse remains healthy. So Griffith decided, well, what if I heat the S cells? What would happen then? And what he discovered was that the mouse was healthy. The mouse lived because the heat killed rendered the S cells harmless. So he decided to see, well, what if I mix these heat-killed S cells, the ones that enabled my mouse to stay healthy, with the R cells that also enabled the mice to stay healthy. So he predicted that the mouse was going to be a healthy mouse. However, that's not what he saw. What he saw was that his mouse died. So what he theorized is that there must be some sort of transformation factor or transformation going on. And he didn't know that the DNA was this transformation, but he knew that something was going on. Because how was it possible that when he extracted blood from these dead mice, that there would be living S cells found in the dead mouse, when before the heat killed those S cells? So he knew some sort of transformation was going on, that somehow the living material from the heated S cells were becoming, I guess, undead, so to speak, and finding themselves in these R strain cells, right? So DNA moved from the heat-killed cells into the living cells, the R strain cells, and ended up killing the mice, thus Griffith developing what he called this transformation property. Now, it wasn't until 1944 that scientists actually discovered that DNA was this transforming principle. So Avery and his colleagues discovered that DNA was the transforming substance. Well, how did they do this? They used viruses as well as bacteria. The bacteria was E. coli, and the virus was a bacteriophage, or just a phage, because from the name, bacteriophage, Phage means to eat, bacteria referring to bacteria. So this virus that they worked with was called the T2 phage, and it killed or infected or took over E. coli. Now we know that viruses don't actually kill the cell. They essentially hijack the cell. So what they did, what was going on here is radioactive labeling of the different parts of the T2 phage. So here you've got the phage head, and within the phage head, you're going to find proteins. Now within proteins, you're going to find sulfur. And within our virus, you can also find DNA. Now within DNA has phosphorus. Now this phosphorus will not be found in the proteins of the phage head. So they let the T2 phage infect the bacteria cell. And here's my bacteria cell. Here's my E. coli. And then they looked at what went inside the E. coli. And what they realized was that radioactively labeled phosphorus was inside the E. coli. Because radioactive phosphorus ended up inside, they knew that DNA must be the transforming principle that Griffith was talking about. So Hershey and Chase provided the information showing that DNA is the genetic information. Our next, important scientist is Erwin Chargaff, and Erwin Chargaff enabled scientists to discover the structure, because now we knew the what. We knew that it was DNA, but we still didn't know what DNA looked like. We didn't know the structure of it. Chargaff enabled the remaining scientists, Linus Pauling, Watson and Crick, Wilkins and Franklin. Those are the three major players that are still in the race to determine the structure of DNA. His base pair rules enabled Watson and Crick to develop their double helix shape of DNA. So Chargaff said that the percentage of thymine is equal to the percentage of adenine, and the percentage of guanine is equivalent to the percentage of cytosine. Therefore, he reasoned that T must always pair with A, and G must always pair with C. So take a moment to review some of these key scientists. Take a minute to see if you can use Chargaff's rule to answer the first question and to look at Hershey and Chase's experiment in the second question. So now we've got Watson and Crick. We've also got Linus Pauling, and we've also got Wilkins and Franklin. Now, Wilkins and Franklin did not like each other. Watson and Crick at this point presented hypothesized a double helix structure. Linus Pauling hypothesized a triple helix structure. So he's out of the race. And now it's down to Watson and Crick and Wilkins and Franklin. So here's Watson and Crick's proposal of the double helix. And when we go to the DNA Research Center or Learning Center, you'll actually see a replica of the model that Watson and Crick developed. So what they said was that you've got this sugar phosphate backbone where it consists of phosphates alternating with sugar. And we know that the sugar in DNA is deoxyribose, followed by our bases, which form the rungs in the middle. Now remember, our bases can have one ring, or our bases could have two rings. Remember pyrimidines and purines telling us which bases have one ring and what bases have two rings? At this same time that Watson and Crick proposed their double helix shape, Rosalind Franklin in England is working on X-ray crystallography, where she's developing pictures of the shadows from the X-ray crystallography that are formed by the DNA. And this is kind of the picture that Rosalind Franklin develops. Well, the story goes that Watson took a visit to England, and because Wilkins didn't really like Rosalind Franklin, he shared her data with Watson and Crick. Using that data and the fact that they were mathematicians, they were able to put together this finalized double helix shape, where the sugar phosphate chains on the sides are like side ropes, and the pairs of nitrogenous bases, one from each side, one from each strand of this so-called side rope, form the rungs. So let me show you what I mean. So here we've got our phosphate, and here we've got our sugar. And this forms the backbone of one of the strands in the double helix. On this side, we've got our backbone, which also includes phosphates and sugars alternating. That is our backbone. Inside, we've got what's called our 3 ring rungs, where we know that thymine and cytosine are 1 ring, they're pyrimidines, whereas adenine and guanine are 2 rings, they're purines. And so there was always this 3-ring rung. 1, 2, 3 rings. 1, 2, 3 rings. Furthermore, the actual nitrogenous bases are held together by these weak hydrogen bonds. Now, why do these hydrogen bonds need to be relatively weak? Well, because they're going to need to be separated during DNA replication. So this was the proposed model by Watson and Crick. Now, the reason why purine and purine could not be paired together, because then your structure would be too wide. You couldn't have a pyrimidine and another pyrimidine, because then your rung would be too narrow. So due to this X-ray data that Watson got his hands on, they reasoned that it must be a purine paired with a pyrimidine. So in addition, Watson and Crick got a Nobel Peace Prize, and Rosalind Franklin died of cancer. Lastly, Watson and Crick discovered that each gene has a unique order of these nitrogenous bases, of this A, T, C, and J.